Hello, everyone, and welcome to Talk Nerdy. Today is Monday, April 22nd, 2019, and I'm your host, Cara Santa Maria. I'm really excited about this week's episode, but before we dive in, I want to thank those of you who made Talk Nerdy possible. Now, that includes Pedro M. Rosario Barbosa, Phil Tiber, the zombie drummer, David J. E. Smith, Jeffrey Perez, Gabriel Felipe Jaramillo Gonzalez, Brian Holden, and Jeffrey Sewell. And if you want to hear your name called out right here at the top of the show, all you've got to do is go to patreon.com/talknerdy and pledge your support. All right, guys, this is a really fun one this week. I sit down with Dr. Jeffrey Marlow. Now, he is working on his postdoc, actually, at Harvard right now, and he's doing some really incredible research on, developing new tools so that he can see microbial communities right there in their native habitats on the ocean floor. Really, really cool stuff. He's also a science writer, a freelance science writer. He does a lot of science communication. And even more than that, he is a Nat Geo Emerging Explorer. So much cool stuff there. We're going to talk about some great science. So, I don't know. Why are we waiting? What are we waiting for? Without any further ado, here he is, Dr. Jeffrey Marlow. Well, Jeff, thank you so much for joining me today.
Well, of course. Yeah, thanks for having me.
I am so excited that we're finally able to make this work. We tried to connect previously, and let's just say that when the new OS downloaded on my Mac, none of my hardware played nice with it, and I had to download all sorts of new software and firmware for some of my recording devices. It was a whole pain in the butt.
But we found a way.
We did. We're connected now, and
That's
you
right.
sound amazing. And you are actually in your lab right now.
That's right.
And tell me about which lab you're in.
Yeah, so I work in the lab of Peter Gerges. This is a lab in the Organismic and Evolutionary Biology Department at Harvard, and we do all kinds of things. Pete's really good about mixing the engineering and the science, taking both of those to the deep sea. So there we study microbes. That's kind of my specialty, but other people in the lab study tube worms, studied the kind of electrical components of how microbes and minerals might interact. And then there's a side of the lab that does a bunch of really fancy engineering to design instruments that actually work on the seafloor. So at any given time, any of that might be happening in the few rooms around me right now.
Very cool. So we're talking tools to understand what's going on deep within the ocean, and then the actual kind of basic science to learn more about these microbial organisms and these kind of deep sea, more complex organisms. But I read that you're a geobiologist, but you're working in an evolutionary biology lab. How do those things play nice together?
Yeah, I mean, I think that any scientist will self-identify in a number of ways depending on the context. So sometimes, that might be astrobiology. Sometimes it might be molecular biology. Sometimes it might be environmental microbiology. But to me, geobiology encompasses kind of all of those. It's really looking at how life and our planet interact. And that can be at any number of scales, really. And I'm interested in the micro scale mostly. So how individual tiny microorganisms deal with their surroundings in really interesting ways. So they might feed electrons to iron minerals that they're hanging out with, or they'll just take up methane molecules from a vent that's right next door. So it's really taking a new look at the kind of intimate connection between Earth and life.
Gotcha. So when we think of the prefix geo, like geology, geography, maybe geopolitics, really what we're talking about is Earth.
Exactly. Yeah.
Gotcha.
And mostly in the context of the minerals, but also kind of the fluids and the air and just whatever the environment gives and takes from life.
So one of the other people, probably the only other person that I know personally who works on deep sea microbes is actually an astrobiologist, and his reason for learning about the deep sea is to try and understand sort of life in extreme environments, and so that if we do come across life somewhere else in the cosmos, we might have some idea of how to look for it. Do you work with any astrobiologists when you do the work that you do?
Yeah, I would say so. It really does depend at the kind of the lens you're viewing this environment through, but we often converge at the same locations. So deep sea vents are among the most extreme spots on Earth, where we're throwing all kinds of environmental challenges at these life forms, and they pretty much find a way to do it. This could be similar to what we would expect to see, what we might have seen at the origin of life, and what we might expect to see as we explore sort of the ocean world beyond Earth, like Europa or Enceladus, where these kind of seafloor energy-rich oases could potentially exist.
Very cool. So with your work specifically, you said you tend to focus more on the microbial communities in these deep sea native habitats. So when we talk about, under the ocean, that's kind of like, that's a lot. That's like most of the planet. So,
doesn't narrow it down too much, yeah.
Yeah, we don't really, it's funny because we don't think about that because we're like, yeah, the ocean, that's like one thing, which is so not. But when you talk about under the ocean, like what specific regions are you looking at? Are you looking very, very deep ocean floor? Are you looking at the ocean floor in places like the Mariana, or do you have specific regions that you're focused on right now?
Yeah, that's a really good question because I think the answer is changing pretty dramatically. So I'm mostly interested in the seafloor. People study all parts of the ocean, from the photosynthetic zone up at the surface to the water column. The sort of pelagic zones is getting a lot more attention these days. But to me, the seafloor is where the biggest gradients occur, especially at hydrothermal or methane vents. And because of those gradients, you can come up with all kinds of crazy life forms that take advantage of that. Mostly I study methane seeps, which, as it sounds, are places where methane-rich fluid are coming out of the seafloor.
So that's like coming from deep within the earth?
Yeah, it's sort of a mix from old, like really ancient organic material that has sort of congealed and been processed into goo and now is kind of just oozing out.
It's like off-gassing?
Yeah, kind of. It's like big, gooey organic molecules, but you're taking little bits of it off. So methane is the smallest type of a hydrocarbon. So it's kind of shearing off little bits of these big organic goo molecules. So that's one type. But another big form is from locally produced methane. So microbes that are right there as well, producing the methane, and then it's consumed locally at the same spot.
Oh, so we're not talking about like a vent and methane coming out from inside of the earth. We're talking about methane being produced right there at the ocean floor.
Yes. Yeah. So it's a mix. And we have some tools at our disposal that help us kind of disentangle which one is dominant at any given site, but both play pretty substantial roles in the methane cycle on our planet.
And how do you figure something like that out so deep at the ocean floor? Like, you can't use mass spec, can you?
You can actually, yeah. So that's one of the really cool instruments that the Gurgis lab is developing and is working on in collaboration with others, mostly at Woods Hole Oceanographic Institution. But you can do this on the seafloor, and that's really important because if you collect these gases and bring them up to the surface, things can change. Maybe some of it's consumed, or it equilibrates in a weird way, or you lose some of it. But to actually get the gas and run it through a mass spec on the seafloor, you can start to figure out exactly what you're looking at and kind of the isotopic composition of those molecules. It's pretty cool.
And is that how you know it's methane to begin with? Because I can't imagine you can just see methane and know it's methane.
That's right. Yeah. Not quite that attuned to it yet. Yeah. So if just sort of figuring out the bulk composition, you can often collect some of that and bring it back to the surface and do it there. But the kind of isotopic component to really figure out where it's coming from is done much better on the seafloor.
Gotcha. And of course, when we talk about the seafloor, I mean, we're talking about like crushing pressures, really insane temperatures. Like, what is the environment actually like down there? Because I don't think most people, I don't definitely think about the seafloor on a regular basis. It's not just like one of those thoughts that floats into my head, whereas you probably think about it all day, every day.
Yeah, it's true. So yeah, it's a crazy place down there that is really variable as well. So the average depth is 4.5 kilometers, which is really, really deep. And it's about 1 atmosphere of pressure. So the pressure that we feel here on the surface, add that amount of pressure every 10 meters that you go down. So 4,500 meters is like 450 times the pressure that we experience on the surface. Most of the places I study are more shallow than that, maybe 800 to a couple thousand meters deep, but it's still pretty intense.
So like at what point do you die if you're not in a, I mean, you die anyway because it's just so deep, but like at what point, if you could have just like, you know, if you're scuba diving or something, should you not go that deep?
Now that's a good question. Don't really have an answer for you on that one.
How do you collect your samples right now? Are people in vehicles? Are you dredging the seafloor? Like, how do you go down and get what you're getting or study what you're studying?
Yeah, so we do need pretty nimble control of our sampling instruments. So that means either a robot or a human-occupied submersible.
Okay. So you're not just free float, like you're not swimming down there.
No, that would be tough.
Oh no. Yeah.
The old school way to do it was, as you suggested, to just kind of dredge or kind of drop a coring device to the seafloor and hope for the best. But when we're interested in these really localized dynamics of how the community could shift right next to a seep or a little bit farther away, you need very tight control, often on the millimeter type of resolution. So that's why now using robots that are streaming video back up to the surface is the way to go.
Oh, wow. So not as common that you yourself have to get into a submersible anymore?
Unfortunately not. Yeah, I mean, it's the most fun way to do it, of course, but
I can imagine it's also pretty expensive.
Exactly. Well, surprisingly, the cost is about the same as a robot.
Oh, really?
Yeah, but I mean, first of all, there's only one that is mostly used by the U.S. scientific fleet, the Alvin submersible, whereas there are a handful of the ROVs, the robots. But to me, it's also just more scientifically efficient to use a robot because it can be down there the whole time. Everyone is seeing what is happening rather than just the 2 or 3 people in the submersible. So it really allows for a kind of more team-based and efficient approach. But it's a really interesting question of the best way to explore these remote environments.
Have you ever been able to go down in a submersible?
I did, yes. I think the second month of my grad school experience involved a trip in Alvin. So it was great. I was like, this is, what grad school's like. Every other month, going to head out to sea and, go in the submersible to the seafloor. It has never happened again. That was a once-in-a-lifetime experience so far.
So you're a postdoc now. You continued some of, at least, did you continue the same research questions or at least in a similar kind of vein as the work that you were doing as a graduate student?
To some degree, yeah. So we kind of seized on this idea that there are carbonate rocks on the seafloor at these methane seeps. And they form huge mounds of rock that can be, a kilometer wide, several hundred meters tall. And this is often where the methane is coming out. And it's formed, in fact, because of the metabolism of these microbes. So the question that I was sort of brought in to answer in my grad school lab in Victoria Orphan's lab at Caltech was to think about what's happening inside these rocks. Are they still active? Are there microbes in there consuming methane? We got a hint that was happening. Pretty conclusive evidence that it was happening at one site. And then over the years, I've kind of collected samples from other spots around the world to build out this story. So that's been a long-term kind of side project for the last few years. But the focus of my work has kind of shifted mostly toward looking at microbes in a range of settings, but really focusing on their interspecies interactions. which really determines how they function and the broader impact they can have.
Okay, so we're going to get to that in just a second, but
Yes.
first I have to ask, you were in Victoria Orphans Lab at Caltech. How have we never met? Why are we not like hanging out all the time? I mean, I know you're at Harvard now, so you're kind of far away.
Yeah, we missed the window, perhaps.
I know. What about like other members of the Nerd Brigade, like the crew that come on Talk Nerdy quite a bit?
That's right.
Have
I think there's
you had any crossover with any of them?
Yeah, I went to high school with Phil Torres. And I went on a fun National Geographic reporting trip to Rome with Jason Goldman.
Shut up. How funny.
So there are a bunch of random
Yes, I have heard all about that trip. How funny. So wait, are you, you're involved with National Geographic, right? Are you an emerging explorer?
That's right, yes.
Oh, very cool. Okay. And also we Aaron Pomerantz, who is another Nerd Brigader. He's an emerging explorer as well.
That's right. Yeah, he stopped by the lab maybe a few months ago.
So random. I love it. Oh, that's so funny. And of course, so many of kind of the Nerd Brigade, my core group of friends out here in Los Angeles, actually went to Caltech, but many of them were in planetary science or in neuroscience or molecular biology or chemistry. So they may not have been in similar departments as you. Yeah. Oh, that's very cool. Okay, so back to your research. Okay, so talking about these microbes, basically, tell me if I'm understanding this correctly, because you're really interested in the interplay between the communities of microbes that are living there on the ocean floor and actually big kind of geochemical things that are happening on the Earth, like that they actually do influence one another.
Yeah, that's right, and it's a tough problem to kind of wrap your head around because a microbe is a millionth of a meter across. They're tiny. And the enzymes that are doing these transformations are several times, 1,000 times smaller than that. So where the rubber hits the road, it's a tiny, tiny interaction. And yet they're influencing things on a global scale. It's 17 different orders of magnitude that we're crossing to go from the enzyme to the planet. So figuring out exactly how to connect those dots is really hard. For example, you can have these cryptic cycles that can happen between different organisms where they kind of shuttle things back and forth before you even see them. So you wouldn't measure that on a larger scale, but it's still happening. So we need to figure out the best ways to both look at things on the individual organism scale, but also the global scale. So I think that's the biggest challenge in our field at the moment.
That's fascinating. And so what kinds of specific, what research questions specifically are you working on right now?
So right now I'm trying to develop a way to look at these microbes in their native habitats, essentially. Mm-hmm. So usually when we collect a sample and want to look at the microbes that are there, we stir it all up, we mush them all together and take out their DNA, sequence it all, and then try to kind of piece that DNA back together to infer what they could be doing or even who's there in the first place. But you've entirely lost the spatial context, so you don't know if a given organism is, next to a rock, if it's next to a reactive iron mineral, or if it's next to a predator that's, consuming it.
And I can imagine that historically that's all you could do, right? Because everything just sort of turns to jelly as you bring it up to the surface.
That's right. Yep. And, we just didn't really have the tools to, as you're saying, first of all, keep it coherent, but also once you've done that, to ask relevant and interesting questions. So,
so you're interested in looking at these things sort of in situ, like exactly where they would be found in nature.
That's right. Yep. And we've been developing a way to kind of encapsulate that with a resin so that, we introduce a molecular marker that's built into active organism proteins. So you kind of trick it by adding in this probe, and it goes into the proteins, and then you can stop everything, embed it in this resin so that it's a solidified chunk, but you haven't moved anything at this point. So you've kind of kept things exactly as they should be and as they were in their native environment. Then we can slice it in thin sections, look under a microscope, and do some kind of phylogenetic marker analyses based on the fluorescence of each of these probes, and really see who's there, who's active, and how it all relates to each other.
Oh my gosh. So it's like an ultra sophisticated sort of classic paleontological move, right? Like pouring or like encasing the bones as they lie in, what do you call it? Like in plaster.
That's right.
And then taking the whole chunk out, moving it to the lab and being able to work bit by bit in a different location, but you still have all that spatial consistency, but you're doing it like at the level of proteins.
Yeah, well, at least at the level of individual microorganisms.
Gotcha. So they really are they like perfused? I guess I'm a little confused as to how they're maintaining their shape.
Yeah, so we kind of just slowly flow fluids through the system. So we collect a kind of core of material. You can imagine that like a big straw or something like that.
Mm-hmm.
Bring it back to the lab and kind of add in drip by drip different fluids. So you add it at the top, it slowly comes out at the bottom. And eventually you're replacing the whole pore fluid in this sediment without disturbing the cells.
Yeah, so it is kind of like a perfusion action. Oh, that's really, really neat. And so the spatial configuration remains consistent because the sample is a really clean, tight sample.
That's right. Yep. And because, so it's all coherent. It all experiences these sorts of flows naturally, like with tides or with currents. So there's water moving in and out normally, and we're not doing anything beyond the scope of what they would normally see.
Mm-hmm. That's fascinating. And so once you have that information, what are you actually doing with it?
Yeah, that's the challenge. That's where we are now. And it's a ton of data. So that's the really exciting part, because we can see really on the individual organism scale where they map out. And that's a lot of information we've never had before. But the next question is how you deal with that computationally and how you kind of design the image analyses to get the relevant information, which is what type of organism likes to be next to another type? Do metabolically active microbes cluster together? Seems like the answer there is yes. The next question is why? And that's a little bit more down the road. But for now, we're just developing this approach to see these different neighborhoods as they really exist.
I love that. So really, in essence, what you're focused on at this snapshot in time is technique, so that a lot of research questions can be potentially answered using that technique.
That's right. Yeah, I think that's a constant back and forth in science and in my career. And something I've always, I think we all struggle with or think about is how far to kind of go on that pendulum from technique development to really addressing a specific environment or question we're interested in answering. Because techniques do open doors, but then you kind of, at least I personally, get in the weeds and sort of lose touch with the importance of the question we're really trying to answer. Is it just developing techniques because they're fancy, cool techniques, or is it really in the service of answering an important question?
Well, and also I think that there's a culture and a pressure around active science, especially at high levels in tier 1 institutions, where there really is an expectation that you're going to be answering big research questions, right? And that you're going to be publishing, you're going to be really productive, and you're going to be publishing a lot in really high impact factor journals.
Right.
And so there must be a little bit of it's frustrating, right? Because honestly, developing new techniques, this is how people win Nobel Prizes. This is how entire fields open up to other scientists. Yet it's sort of not smiled upon the same way within the institution, is it?
I don't know. I'd actually sort of propose the opposite. I think that technique development, at least in geobiology, in my narrow field, does seem privileged in a way.
Oh, really? So people really give you a lot of kudos when you're able to develop some sort of new way to investigate a question.
Yeah, I think that's right. not me personally, but I don't think my contributions are quite on that level at this stage. But the ones who are developing some of these really new ways of seeing the world, initially you maybe question why this matters and why this is worth devoting so much time and effort to. But I think the proof kind of comes years and decades down the line that it's probably worth it. And I think that, a lot of the reward structures are starting to recognize that.
That's good to hear, because I would fully agree with that. And I think it comports well with what I was saying, that sort of sometimes in the moment you don't quite get the support that you might need from your, not from your institution, right? Your institution hopefully is supporting the work that you're doing, but from the community at large. But that later on down the line, like, these are the people who often are getting awards, or their work is really going down in history. The reason I bring this up is because I just had a paper due like last weekend for my history and systems course that I'm taking, because I'm working on my psychology PhD. Yep. And the question posed was, does Kuhn's paradigm shift model from the structure of scientific revolutions actually apply to the social sciences? And if so, how does like the fragmented world of psychology comport with the sort of classic examples that Kuhn uses, coming from physics? Because if you've never read this book, it's about how science changes and revolutionary science occurs. He actually coined the term paradigm shift. Right. So, you can tell that it was that important culturally. But he used physics examples. So they were very clean examples, like the Copernican revolution and like, Galileo and Einstein's relativity, like things where you can be like, yes, the world is different to us now than it was before we knew these things. But one of the points that he makes a lot, and that's why I bring this up, is about the fact that technology is sort of the key. In a lot of these cases, we couldn't know what we now know, and we couldn't see the world differently if certain types of, certain pieces of technology weren't developed. Right. If we didn't have, think about gravitational wave physics. It's a whole new type of physics. It's a whole new type of astronomy. And prior to being able to measure them, we couldn't even prove that they existed.
Yeah, totally. So do you see that in the social studies context? Like, are there, sort of analogous method development progress that sort of unlock new ways of seeing things in the same way?
Early on in psychology, it was all about, there was a whole movement that was all about tools. There was like the classic psychologist lab back in the day was all about reaction time studying and things like that. This was like hundreds of years ago, or 150 years ago. Now I'd say it's probably much more about, it's less about tools and more about statistical techniques that can really open things up a little bit. Because of course, there's only so many ways to study mental phenomena or behavioral phenomena. There are periodically new tools that open things up, but I think the question really is, and the point that I made in the paper is that psychology is fragmented and will probably always be fragmented, and that's not a bad thing. Right. Like, the same rules don't need to apply to physics as they do to psychology. But one point that Kuhn makes in the book that I think a lot of people don't notice is that he says that a scientific revolution, a paradigm shift, doesn't have to be so global as to affect every single person in the umbrella field. It can be as small as to affect 25 people that have been working on a very particular research problem. So in that way, I do think that we see many paradigm shifts all the time within social science and really every other field, where something new happens within a very particular area of research interest, and all of a sudden those people see the world in a different way and they can't go back. Because that's the whole point of the paradigm shift, is incommensurability, that it's not a gestalt shift. You can't see it one way and then the other way and then one way.
It changes fundamentally, kind of.
You cannot see the world the way you used to see it now that you've introduced this new information or this new data. So have there been any big paradigm shifts in the work that you do, in microbial geobiology?
I think that I'd maybe point to a couple, and, back kind of the idea of methods versus kind of system-specific questions. I think on the method front, just the ability to sequence everything has changed everything. I never was quite at the stage of kind of reading gels of DNA sequences line by line in an electrophoresis gel. But
wait, you weren't?
Not quite.
Wait. Well, you're a postdoc. How long have you been a postdoc?
4 years.
I read gels. Okay, 4 years. I read gels when I was in college. I mean, I wasn't a researcher, but definitely in lab we read gels.
Sure.
But I guess you don't have to do that anymore, do you?
I think I barely was after that, but
oh my gosh, I remember mixing the auger and working with the student, because I used to teach bio lab, and putting the different dyes with different molecular weights down into the little wells and run. Oh my gosh, there were so many supplies you needed.
And then you enter the dark room and put on the UV light and hope for the best.
Oh yeah, we didn't use the fluorescent gels in bio lab. Yeah. That was like actual science. You would use those, but when you're just demonstrating with the bio students, you just use really bright dyes. Yeah. Nice. Anyway, so now everything's done with these tiny well, maybe not tiny, but there are there's developments of these tiny portable sequencers, aren't there?
That's right. Yeah. So I've never actually used that. Because it's,
that's like the work that Aaron does in the rainforest.
The mid-ion systems.
But you don't need that because you're not actually taking it under the water. You're taking your samples up and back to the lab.
Exactly. Yeah, you do pay both financially and kind of accuracy-wise for the miniaturization.
Sure.
It's worth it in a lot of ways. But when you can just send stuff to a core facility and they'll sequence hundreds of thousands of different organisms for $100, it's hard to argue with that. So,
and so when we say sequencing organisms, just to interject a little, we're talking about taking, like you mentioned, taking that DNA out of the sample or RNA, like however you're going to measure it, and actually comparing the base pairs to a giant database and figuring out which organism they belong to.
Yeah, that's right. So it can be DNA, it can be RNA, or it can be proteins. So I've mostly...
you can do it based on the proteome.
Yeah,
also.
Exactly.
Okay.
So I did a fair amount of work with the proteome at one point, and that's again the molecular machines that are really doing the work. So in some ways, the DNA is telling you what's possible, but the proteins are telling you what's really actually happening. So it really just depends on the question you want to answer and kind of the best way to preserve and process your samples.
Very cool. Okay, so, and this has now gotten to the point where it's cheaper, it's easier for certain people who need the capability for it to be miniaturized. It can be miniaturized.
If you're going to the space station, for example, they just did that recently.
Like completely different than it was even just 15 years ago. And of course, looking back a few more decades, I mean, PCR really changed things, didn't they? Because it used to be you couldn't amplify the actual DNA. So you were stuck with testing the teeny... I mean, you probably wouldn't have been able to test microbes without PCR, would you?
No, not much. Or we'd have to extract from huge quantities of material. So normally, right now, I can take about a gram of soil or sediment from anywhere, mush it all up and get plenty of DNA to sequence with PCR once you amplify it several orders of magnitude. But so if that weren't possible, I'd probably be dealing with liters of the sediment to extract enough DNA to do anything with.
Oof. Yeah, so that's polymerase chain reaction and just like quick and dirty is it actually, like you said, amplifies. It's going to like take a little bit of material and make it, I guess, how does PCR work actually? Do we know the answer? I'm sure. I'm like somewhere in my brain, I know this.
Oh yeah, no, tell me about it. Essentially uses a enzyme that operates at high temperature to replicate DNA strands. So you will add in little primer sequences. So if you know the sequences of the ends of the chain that you're looking for, you can add in those little bits that attach. Then you add in this enzyme that goes and adds the different A's, T's, C's, and G's to this growing chain. And because it has a template, the initial strand, you'll get a copy of the complementary strand.
Gotcha.
And then you can repeat that over and over until you have all the DNA you need.
All the DNA you need. And talk about paradigm shifts. I mean, PCR really changed the game. I mean, it changed the game the way that I think our generation looks at CRISPR.
That's right.
that generation really looked at like, wow, we can do molecular biology cheap and easy. Like we used to never be able to do it to this extent. And that was really, just a few decades ago.
That's right. And because of that, it brought new low biomass extreme environments kind of into the realm of the living. Before we had the ability to make these copies, you maybe wouldn't have seen any life. But now we're finding that pretty much anywhere on Earth that has liquid water, there's something. So I think that's been one of the other big paradigm shifts in terms of our understanding of biology, is that life is kind of everywhere. In the deep sea, these chemosynthetic, chemistry-driven environments, it's separated from the surface world to a pretty impressive degree. So that's been, to me, the most exciting part from the science side.
All right, everyone, I want to take a quick break to thank the sponsors of this week's episode, starting with EveryKey. And I don't know about you, but I have a really hard time remembering the passwords and the logins to all my different accounts, all my different devices. And honestly, I don't have much time in my life, and EveryKey actually helps with both of those things. It makes my life less frustrating because it's a single device that ensures that I never have to remember another password or login again. It's actually a Bluetooth key that unlocks all of your devices for you, your laptop, your phone, and your tablet when you're close by, and then it locks them back down again when you walk away. And soon it's going to unlock your car and your house too. It can also generate new secure passwords for you. No more sitting there on the spot trying to dream something up. And it remembers all your current passwords and logs you in automatically. And get this, if you ever lose it, you can easily freeze it to make sure that no one else can use it. It's huge. It's just makes life so much easier. No more time wasted typing passwords. No more worries about hacks. It unlocks time. It unlocks your peace of mind. I'm going to be honest. And right now, if you visit everykey.com and you use the promo code NERDY20 to purchase EveryKey, you'll get 20% off. So remember, that's everykey.com with the promo code NERDY20 for 20% off. I also want to thank Landish for their support of the show. Landish makes nutrient-dense and sustainable food bars and protein powders. And guys, these are perfect for those crazy days where you just need to throw something in your bag that you can eat later. And you have the option of bars or powders that are made with really cool ingredients like spirulina, which is a spiral-shaped microalgae, actually a cyanobacteria, reishi mushroom, which research now shows has beta-glucans, which are polysaccharides that, might be actually immunostimulating. And lastly, I think this is still my favorite, cricket powder. They're milled crickets, the actual insects, yes. They're, no lie, free-range farmed in Canada. And the truth is they're really nutritious and they're incredibly sustainable. So you can learn all about these ingredients. You can learn all about their nutrition facts on the Landish website. But let me give you some highlights here. They're macronutrient balanced, so they've got lots of protein and good fats and fiber and not nearly as much sugar as, most of the foods that we're eating right now. They're micronutrient dense. So there we're talking about things like vitamins and minerals. They all provide some form of prebiotic fiber, and they're actually really tasty, because that's pretty dang important. They're not overly sweet. They're not chalky. It's a different kind of protein, and I think that you guys are really going to love them. Right now, you can get a big 30% off at landish.co by using the promo code NERDY at checkout. So that's 30% off if you go to landish.co and use the promo code NERDY. All right, guys, let's get back to the show. So you mentioned low biomass. I think that I've always been of two minds about the deep, deep ocean floor because historically, I think people believed that it was just dead, that there was nothing down there. But then we discovered that there are these chemosynthetic organisms. And I guess my perspective shifted having seen so much research and so much writing about it that I'm like, oh yeah, it's just like teeming with life. But is it sort of compared to, a coral reef or something like that? It's actually still pretty not dense when it comes to biomass, or is there actually a lot of living stuff down there?
There is, yeah. So it certainly depends, of course, but the most productive hydrothermal vents, for example, are about as dense in terms of biomass as the rainforest.
Shut up.
Yeah,
oh, that's cool. It's just that everything's really small.
Everything's small. It sort of goes probably a little bit deeper into the subsurface. But there's kind of just as much coverage and biodiversity there. The background seafloor, it still has, maybe 1 million cells per gram of soil. So that's a lot, but it's 100 or 1,000 times less than what you'd see at a hydrothermal vent.
Gotcha. Okay, so just in general, the seafloor is...
it's more barren, for sure.
Yeah, it's more barren. That's the word I was looking for. Thank you for that. I was like, less lifing isms. Yeah, it's more barren than like the soil on the surface. But at these hydrothermal vents, it's still pretty lush.
Yes. But it's interesting that you... I think the common perception of the seafloor maybe is that it is full of these dramatic hydrothermal vents, because that's often what we see expressed in the media. And this all, it's all a positive feedback loop, but also because it's where most research expeditions go. We don't really have a great sense of what kind of the background normal seafloor is like, because we're often going to the exceptions rather than to the rule.
Oh, that's an interesting and important point to make. And one that you're right, I hadn't really thought of. And I can only assume that these hydrothermal vents are not just like everywhere.
Right.
Most of the seafloor is just seafloor.
They're really quite rare. Yeah.
It's interesting that you bring that up, and I don't want to shift the conversation too much, but you obviously are dialed into this, I don't want to call it a problem, but this interesting point that you made, because you yourself are an active science communicator in addition to doing the research that you're doing, and you've written for some really great outlets before. And one of the things that you wrote just recently actually was a piece for The New Yorker all about what happens when a massive organism, a whale, right? A huge part of the ecosystem of the ocean, dies and falls to the seafloor. So I would assume that the ecology surrounding, because it is kind of like its own little mini biome, right?
Yeah.
The ecology of a dead whale is probably one of the more biodiverse parts of the seafloor.
It might be. Yeah. I think that we're still trying to figure that out. The scientists who study these environments in particular. But so, yeah, I think it's absolutely would be one of the most sort of biomass-rich spots on the seafloor. But biodiversity sometimes takes time, because you need to start to degrade those like primary materials and allow for all of the feedback loops and kind of consumption of products to then feed back to other organisms. So I think because these are, evolutionarily speaking, flashes in the pan, they maybe last for 50 years. It might not allow for that degree of biodiversity to sprout up.
I love it. It's transient, even though it lasts for 50 years. Like, I want to unpack that for a second, because you're right. From an evolutionary perspective, that's nothing.
That's nothing, yeah.
You might not even be able to measure that in the fossil record. But from kind of a gee whiz perspective, like, I guess it makes sense that it would take that long for an entire whale carcass to disappear, or to be, utilized. But 50 years sounds like really impressive. Like, can you imagine if on the surface it took 50 years for just like a body in a forest to become bone? Well,
yeah, it's interesting. I think it has a long tail, I guess I would say, in the graphical sense. The flesh is eaten pretty quick. So I think... Okay,
so it's similar, like on the surface.
Within maybe, 2 years, it would be...
That's still a really long time.
It's true. That's true. But then the next 48 is mostly like bone-eating worms and other things that kind of sprout up, because the primary producers have done, or the first scavengers at least, have done the first bit of that work.
And so we're talking like the actual carcass is gone after 50 years. Like, there are no bones left.
That's right. Yes.
Okay. Okay.
Yeah.
Yeah, I don't know. Do we have bone-eating worms on dry land?
Yeah,
Because I feel like bones last forever.
So. Yeah,
I don't think so either.
They're called ossidacs on the seafloor. They're these really crazy little things that have, they bore down into these bones, and their symbiotic microbes do the work and then sort of feed the products to the worm that hosts them. And I'm pretty sure they were discovered on the seafloor in whale bones. And then whether or not that extends to other environments is really just, starting to be examined. But it's one of the, I think, very few cases where the first example of something is found in this really extreme, bizarre environment. And now we're looking at our more prosaic environments to see how common this actually is.
Ooh, that's really cool. And that happens quite a bit in science, doesn't it? Where, and sometimes you never find the thing outside of that really extreme environment, but other times you're like, oh crap, this is like everywhere.
Yeah.
How did we not notice this before?
Exactly. Yeah, there's a cool organism that this Danish group discovered that kind of transports electrons over kind of centimeter scale distances, which, given the fact that a microbe is a millionth of a meter across, that's huge. That's like thousands of body lengths that you're kind of communicating with similar cells. And I think they initially found it maybe on the seafloor, but it's found all over the place. So you'll see people going to sample like drainage ditches outside of their offices and their backyards and finding the same crazy organisms.
Very cool. Oh my gosh, that's so neat. It's definitely an interesting thing that that extreme environments can often be a template for things that we understand to be existing all over our own planet. But like we kind of talked about before, that we only really touched on it at the beginning, that the biology in extreme environments on Earth can actually be a potential template for trying to understand what life might seem like if we were to encounter it in elsewhere in our own solar system.
Yeah, exactly. I think that by figuring out the limits of what Earth life can do, that really shines a light on what the limits are and what life really is. I think, you can't really define something necessarily until you've seen the far reaches of its capabilities. But then the link to other worlds becomes a little bit more theoretical, because we're still just starting to figure out what those environments are really like and how we would actually go about getting data from those spots.
Yeah, absolutely. And I think that it's a little more, I used to think, how could we even potentially look at someplace like Europa or Enceladus and think that we would know how life would be produced? But then the more I kind of learned about basic chemistry and physics from doing science communication, because those were not my fields of inquiry when I was, going through my academic career, the more I realized that the building blocks are generally the same. There might be some bizarre stuff that we discover, but the basic rules probably won't be broken too extremely, simply because we're all made from the same stuff in the Big Bang and then in the furnaces of these big stars. So that kind of helped me realize, you don't have to throw the book completely out and start from scratch. There are some restrictions.
Laws,
yeah.
I think that's
which is good, because I think that that's really, one of the things that I'm working on with my advisor right now is an area of research. It's not really my research area of interest. It's not going to be my dissertation or anything, but it's like a side project. It's all about creativity. And we're often in psychology, there's a lot of argument about what creativity even is. How do you even define the construct of creativity? It's actually a pain in the ass to study, if I'm going to be honest, because what the hell is this thing? But one of the things that I have always argued for is that creativity requires constraint, that it's not a creative endeavor to just do anything and everything without any sort of rules, right? One of the ways that we measure creativity in young children where there's less constraint is we might give them a tool like a paperclip and say, what can this become?
Right.
And then as they get older, we actually care about whether the paperclip looks like the thing they're saying it is,
Right.
or actually could be functional. Like you can't just take like a mug of coffee. I'm looking at my mug of coffee. See, this is me not being creative. And I'm like, it's a rocket ship.
Sure.
Like, okay, in my imagination, sure, but like nothing about that mug of coffee is like a rocket ship. And so once you have those kinds of constraints, you might actually be able to utilize creativity to solve real world problems.
Right. But do you think, I mean, in the case of the paperclip, at least, the constraint is kind of, societally imposed. It's not a actual
Absolutely.
constraint.
Yeah, and that's why too I think that those changes to how we judge it change with age.
Definitely.
Because littles don't, they're not victimized, I guess, by societal imposition. Yeah. And in a way, that's why we like to say, look at the world through a child's eyes, right? That's how scientists should approach problems. But at a certain point, knowing whether or not something is going to work is also important. Having read the literature and understanding how not to reinvent the wheel is also a really important part of the scientific process. So, all those things really do come together.
Yeah. I think it's healthy to bump up against both the kind of culturally imposed constraints and also the physical constraints to push the field forward in both ways.
I agree. And knowing the difference too, because I think if we're not, that's one thing that I do really love about the social sciences that I did struggle a bit with when I was in a pure neuroscience lab, is that the contextualization of, like you said, like society, race, gender, just these cultural norms, they actually do have a lot of interplay. But I think in the hard, quote unquote, hard sciences, we tend to just pretend that that's not the case. Whereas the social sciences at least kind of recognize that to some extent. And they train, their graduate students in cultural competency and multicultural views and things like that. It's something we got to catch up on a bit in the natural sciences.
I've actually been thinking a little bit about this interaction of the social and natural sciences, and I could take a real psychologist now that I have you to
working on it
to answer this. But this question of how we kind of explore the most remote places of our solar system, whether that's space or the deep ocean, when we do it in person, whether if you're an astronaut or you're in a submersible. As opposed to doing it through telepresence, through a screen, essentially, how does that really affect the nature of the science? It certainly broadens the type of and number of people that can participate, which is amazing. It really democratizes it, but does it change like the features we find salient or the the specific questions we're asking?
Yeah, I mean, you think about it and you would think that it must, simply because it actually does have an impact on the kind of awe factor, right?
Definitely.
Like being there in person, seeing it with your own eyes. Then again, there's only a handful of people who get to do that.
Right.
And, being able to share their tale, I think, is really important. Being able to see it with your own eyes as somebody who maybe would never be able to go, but looking at it through a screen, you definitely have the capability. I mean, think about the moon landing, for example, and how amazing, how just incredibly earth-shattering that was. But for kids today, they watch a videotape of it and they're like,
exactly. It's still something on the screen.
Okay.
Right.
Yeah, exactly. And even still, it's partially because it's something on the screen. It's partially because it just doesn't feel new anymore. Like when... I think that when our generation watched I'm lumping you in with me, deal with it, watched Curiosity landing in Gale Crater, we were like crying, and it was like a really big deal, especially to the nerds. I'm not sure how much it had global appeal, but I think it had pretty big appeal.
I think so,
But then since then, we've had other, and we're like, okay, yeah, that's another one.
Yeah. Yeah, it's funny. Every time there's going to be another Mars landing, it's such a hard calculus, I would imagine, for JPL's PR team to run between this is so dangerous, it could crash at any time, versus we are very competent and are confident this will work.
Yeah. And also, like, I know you feel like you've seen this before, but trust me, this one's new.
Exactly.
I think that's a big concern, right? So people are really excited about putting boots back on the ground on the moon. They're definitely excited about putting boots on the ground on Mars. But it's something I argue with on my other podcast, The Skeptic's Guide, all the time, because I think it is a massive waste of resources to put people on Mars. And I don't see how it's ever ultimately going to be worth the risk-benefit analysis. It's so incredibly dangerous. And just for the sake of going, ooh, I'm just not sure it's worth it when we can do really good science remotely. I'm not sure it's worth the risk to human life. And so I think that, there's a human spirit of exploration. We know this. We both have, deep ties to National Geographic, for example, which embodies that exploration spirit. But I also think that exploration should be, done smartly, and B, now we're understanding, it only took us a few thousand years, the real impact of colonialism on exploration and doing it in such a way that it's sort of healthy and doing it in a way that it's respectful. And I really do see, people have argued with me tooth and nail because they think I'm being like a hyper feminist, but I really think like man go to Mars is just a new example of this like deep entrenched colonialist. Yeah, exactly. That I think we have to look at again.
I think that's right. Yeah. And ideally all of these endeavors are done for a purpose and to answer a question.
Exactly.
And if we can do that with a robot, then let's do it with a robot. I think that once the ego and less savory cultural components come into play, or going down the wrong path anyway.
Yeah. And I mean, I think that at a certain point, just doing it to do it, like you look at somebody like Alex Honnold, like he really wanted to free solo El Capitan. I just watched the film recently. If you guys have not seen Free Solo, please see it. It's like, it's intense. But like the whole time you're really feeling for, his significant other, and you're really feeling for the camera crew and like the intense psychological pain that they're like, this guy could die and I could be filming it. And like, I could be a party to that. I would feel guilty for the rest of my life, I mean, obviously it was his choice and nobody was forcing him to do it and all that good stuff. But just for the sake of saying, I've done it, and once you start to actually put other people at risk, when you're an institution putting somebody on a ship, that calculus really changes.
That's true. Yeah, I think that, I mean, maybe the last, few decades, the separation between adventure and exploration, I think, have kind of become more apparent. Like, 100 years ago, if you wanted to answer some of the most intense scientific questions and needed to like go get a penguin, you had to go on this really intense journey to get there. But now we can get the highest resolution images of Mars from your computer. So they used to be, I think, a little bit more intertwined, and they really don't need to be anymore.
Absolutely. And I think that now we have the tools and the wherewithal to know how to do these, exploration does still exist and it still should exist, I think. And, we really still have fundamental questions that we're trying to answer, but we have to do it in a way that doesn't negatively impact indigenous communities. We have to do it in a way that is not a waste of resources. We have to, like, all those things matter, and we're finally like aware of that. And it's sad that it took so long, but
absolutely. Yeah. And in a way that's, inclusive and not dictating what people halfway around the world are finding, but we're all on this journey of exploration together, and you can do it. The idea that you can find the craziest new microbe in a drainage dish outside your house means that the frontier of exploration is anywhere that you kind of make it to be.
For sure. And I think that's why citizen science is such a cool thing. And it's so cool to see so many citizen science projects popping up everywhere. And guys, if you're not involved in a citizen science project, or you don't even know what that is, I highly recommend that you start looking into it, because there's a ton of ways. I mean, there's like intense ways you can be involved, like putting in, malaise traps in your backyard to catch bugs and learn about the biodiversity in your community. And then there's like go online and sort galaxy pictures, and like gamify it and spend a few hours on a weekend when you're bored, contributing to science in that way. So it really does run the gamut. Yeah. But you're right. Like it's so democratized now. But on the flip side of that, there still is a lot of exploration to be done. Oh, I was going to say, sorry, you could tell that I was trying to buy my time while I was figuring out what I was going to say, because I remembered and I was trying to, or I forgot and I was trying to remember. Also, there's this concept that science writers, and I think science communicators have to be, we have to be better. So when a quote unquote new species is quote unquote discovered, really, was that species named by science? Did indigenous communities always know about this? It's one thing when we're talking about microbes, sure, but like there are times when we're like, oh look, a new great ape. It's like, it's not new.
Yeah.
we just hadn't come across it before out here in the West, And so having that kind of cultural competency to talk about these things in a way that's respectful of cultures that maybe just didn't feel the need to share it with the rest of the world, is also, I think, important.
Yeah, this is something we saw changing gears a little bit, but I was just at the United Nations talks on coming up with rules around biodiversity on the high seas, and written into just about every component of this in terms of how we're sharing knowledge, how we're equitably sharing the resources that come out of these areas was an explicit inclusion of indigenous knowledge, which is really great to see.
Amazing. Well, and also duh, It's so helpful, right? Like we've learned so many things. Like I think a very particular story that I don't remember the details of having to do with narwhals comes to mind that that scientists couldn't find a narwhal population when they were trying to track it. And Inuit communities were like, well, yeah, we track them every single year because they're they're central to our way of life here. Sure. And here, we know all this stuff. Would you like to know
just
it? And they're like, holy shit, thank you. So yeah, it's obviously now, we can work together and solve these problems in a better way. Yeah. So I'm glad you brought that up because that's kind of one thing that I wanted to discuss here before we close out the conversation is that not only are you involved in active, science and science communication, but also one thing that I really respect and appreciate is that you really do care about policy. One of the things that I've noticed a lot lately is a sort of backlash or a sort of controversy around a scientist, you should stick to your science or don't advocate or no activism from you, scientist. Right. And I personally think that science and policy are inextricably linked. And so it kind of bothers me when people have this mentality that scientists shouldn't have a political stance.
Yeah. I mean,
because we're informing the politics.
Exactly. I think, there's a lot there. I think that science should be able to accommodate anyone who wants to be involved. And for some people that is, dealing with the exact flight path of a ion through a mass spec, and that's all they want to do, and that's great because that's really important and it allows for a number of questions to be answered. But other people,
I don't know what those questions are, but someone out there does. So.
That's right. How much methane is there?
There you go. Okay, yeah. This is fundamental to your research. Sure.
Sure. Okay. Yeah, that's true. But so I understand when people like that who have spent their whole career getting great at that only want to do that. That's fine. But if there is a subset of scientists who want to engage with the public, whether that's through writing or in policy or through activism, I think that's all the better as long as it's kind of rooted in evidence-based reasoning. On the policy side, we're mostly there to kind of support the negotiations. And if any scientific questions arise, we're conduits to a network of scientists around the world who can get the best answers to inform the policy. It's not, at least in the oceans context, particularly advocating for anything just yet, other than what everyone's there for, which is to conserve biodiversity in the high seas.
So we're really talking about these areas that nobody has claim to, so multiple constituents are concerned about ensuring that they are conserved and respected.
That's right. Yeah, so it's the areas beyond national jurisdiction. So every country has their exclusive economic zones out to about 220 miles from their coastline, but that leaves 45% of planet Earth unaccounted for. So that's a lot, and it's pretty remarkable to me that there aren't really comprehensive laws designating what happens there and how to protect those areas.
Yeah, and of course, these are places where things unsavory things can happen. Like dumping has obviously historically occurred quite a lot and probably still does. And just like things that aren't very good for the life that is living in our oceans.
Yeah, absolutely. And all kinds of fishing. There are really cool new satellite-based approaches to kind of track fishing vessels and see if they're in compliance with their country's laws. So another case kind of where the tech is starting to meet the challenge.
And so you wrote an op-ed a while back about the idea of this kind of conservation effort, the exploitation of these areas known as the high seas, and how maybe our science and our exploration hasn't really kept up with the utilization for commercial reasons, and that there's a disconnect there a little bit.
Yeah, I think that's right. It certainly hasn't kept up with anthropogenic influences in general. So when, the second we started emitting CO2 or other pollutants into the atmosphere, it was inevitable that would reach the far reaches of, ocean trenches because the atmosphere circulates and water circulates. Yeah.
Yeah, and this is people on land. Like,
right.
This isn't people who are going out to the deep sea and like actively doing things that are negatively impacting it, because the Earth it's a one giant biosphere.
Exactly. Yeah. Yep. So we at the time, didn't really know that was the case, but in the intervening centuries, it's certainly gotten everywhere. And even before we start to explore some of these areas and find the amazing biodiversity that's there, we've already affected them, and we're probably already killing off some of the species that we haven't even kind of found yet.
Ugh, I'm getting sad now. Yeah. I don't want to get sad.
Sorry.
But what you really say in this op-ed that you wrote for Undark is that we need to keep up with, we need to explore so that we know we can understand the pace of this, this detrimental activity, whether it be climate change, ocean acidification, all these things that are happening from our global activities to, like you said, this overfishing or dumping or plastic pollution or, whatever the case may be. Like, we need to understand the extent of this stuff with scientific investigation so that we can mitigate it.
Absolutely. Yeah. And I think that's where the sort of less centralized, whether that's through telepresence or through autonomous vehicles, that's where it's really going to help us. So, yes, it's great to go to the seafloor and be a submersible, and it might change your life, but we're not going to cover the scope of the problem if that's all we're doing. So I think that's, really making the case for going out to explore new environments, not only going back to the same hydrothermal vents or methane seeps to do the same types of experiments, but to get out there and see what's going on.
Yeah, it reminds me quickly before we get to the closing questions. It reminds me a little of an earlier episode that I did here on Talk Nerdy with I think her name was Jo Ruxton, and she was a producer on Blue Planet.
Okay.
And one of the things that we talked about was that after doing Blue Planet, she really felt the need to do another film. Obviously wasn't nearly as big a film, but it was, I think, in some ways more impactful, called A Plastic Ocean, because she felt like while she was working she's a director too, actually. I think she directed Plastic Ocean. But while she was working as a producer on Blue Planet, or maybe a director on that too, I might be getting my wires crossed, she realized that so many of these, beautiful, pristine shots that they were putting forth as sort of an aspirational part of the documentary, or the whole documentary is aspirational, were simply not reflective of reality. So, you'd be doing this beautiful pan, and then you'd have to cut the tape because you'd see garbage, or you'd go to one place to film and you'd be like, oh, we can't use this. It's too depressing. And so you'd go try and find these really cherry-picked samples of pristine ocean, which are not reflective of the ocean. So she actually felt guilty presenting that and wanted to follow up with sort of a documentary of what the oceans actually look like. And I think that's an issue that we're all dealing with in SciComm, is how do we talk about these conservation stories so that we're not depressing people, but we're inspiring them, but at the same time, we're being honest.
Right. Yeah, is the job to kind of share the wonders or the reality? And yeah, I don't know. That's tough.
Yeah, and maybe it's both. And
Absolutely.
maybe we've just got to make sure that those things are balanced. I mean, obviously, that's the new Attenborough docuseries really is all about that. Yep. It's like finally, after, decades of the sort of aspirational work that he's done, I think he felt a moral imperative to talk about some of the detrimental things that are happening to the planet. All right. Well, Jeffrey or Jeff, you go by Jeff.
Yeah.
Jeff, sure. Jeff, we always close the show with the same two questions. And definitely as somebody who's working in these really extreme environments and looking at these very, I guess, fundamental organisms, I think that's a good use of the word fundamental because they could really unlock a lot of secrets about, life on this planet. I'm definitely interested in your take of these final questions. So, to start, when you look to the future, what is the thing that is most, disconcerting for you? What keeps you up the most at night? What are you actually, legitimately concerned about? And then on the flip side of that, what are you genuinely kind of hopeful and optimistic about?
Yeah, I mean, kind of what we were just talking about, that we're really not so attuned to the long-term effects that we're having on this planet. And to think that these amazing discoveries are kind of slipping through our grasp before we even get a chance to make them is pretty concerning. So that's not great. But I do think that the technology, at least, is getting fast enough and getting high throughput enough that we don't need too much time to catch up, that we can find out the fundamentals of biology, at least, in some of these extreme locations before they disappear. So I think that's what's encouraging. The open question, though, is what we're going to do with all of that knowledge on both sides. Are we going to minimize the harm and maximize the benefits of these discoveries? And I guess that's the neutral, open-ended question for the future that I'm hopeful we will address in the right way.
Oh, me too. And obviously the work that individuals like yourself and your colleagues are doing is really important to be able to mitigate and even potentially start doing more than just mitigation. Right. So I thank you for that. I think the people listening thank you for that, and we're definitely interested to see what comes next. Jeff, how can people follow some of your work, maybe your SciComm work, but also some of the science that you're doing? Where can they find you online?
Yeah, I think the best clearinghouse would be Twitter. So I'm at JJ_Marlow. I like to kind of pinpoint cool work my colleagues are doing, interesting things I found, but also, some of the stuff I've been seeing and doing in the lab.
Very cool. Okay, great. And of course, I will link out to some of these articles that we mentioned within the show notes so that people can take a look at some of the SciComm work you've done in the writing as well. Jeff, thank you so much for joining me. This was fascinating.
Absolutely. Thank you so much for having me.
Of course. And everybody listening, thank you for coming back week after week. I'm really looking forward to the next time we all get together to talk nerdy.